And welcome back to you as well. Indiana's life sciences industry is known for its collaborative efforts, both in our own backyard as well as around the world. The next section of our conference focuses on what we've learned about partnerships and working together over the last 18 months. So to start things off, we're honored to have Dr. Anthony Philippakis. He's Chief Scientific Officer of the Broad Institute in Cambridge, Massachusetts. At the Broad Institute, he's founding director of the Data Sciences Platform, an organization of over 200 software engineers and computational biologists that develop software for analyzing genomic and clinical data. Today, Dr. Philippakis is going to share how COVID has impacted how the Broad Institute approaches research collaborations. He'll also give us some ideas of what the industry can do to continue that momentum. Dr. Philippakis, welcome.
Thank you so much. What an incredible opportunity to be here today and participate in this incredible event. I'm sorry I couldn't be there in person. Maybe will you start the slides now? Is someone going to show my slides or? Oh, they're there. Okay, great. Next slide. So, as you mentioned in the introduction, I wear 3 hats. By training, I'm a cardiologist, trained at Brigham and Women's, don't practice anymore. Most of my time is spent at the Broad Institute, where I'm the Chief Data Officer and lead a number of efforts related to software and data science. And then finally, I'm a venture capitalist. I've been a partner at Google Ventures since 2012 and work to build companies and make investments. So the talk today is really about my work at Broad, but I think you'll see echoes of the other 2 areas. Next slide. So as we think about highly transformative and collaborative projects that go on around the world, I like to break it down into 2 axes of risk whenever you're starting a new initiative that could be potentially very impactful. So on the one hand, you have execution risk, and on the other hand, you have intellectual risk. So if you think about that bottom left corner, that's low intellectual risk and low execution risk, I'll be honest, I haven't found too many impactful projects that actually have neither of those characteristics. If you find one, good luck, go after it and grab it quickly, but I've never found one. But let's think about things that have more execution risk. And here, I tend to think about a lot of large-scale human subjects research activities. For example, all of us, an effort to recruit 1 million Americans, get full medical records on them. Similarly, UK Biobank, its equivalent in England. These are efforts that don't require new ideas. Subjects research studies is really quite a difficult task and one not to be taken lightly. So against that backdrop, it's also interesting to consider the other extreme, which is projects that have large intellectual risk and low execution risk. So these end up being serendipitous scientific discoveries that change the face of medicine. Things like the discovery of siRNA that allow us to silence genes, or more recently, the discovery of CRISPR. And then finally, there's a class of projects that are actually both high execution risk and high intellectual risk. And those are the ones that are often most impactful to society. My favorite example is the Human Genome Project. In order to sequence the human genome, we had to have a whole new series of intellectual breakthroughs in new sequencing technologies, new algorithms in order to assemble the genome and annotate it. Similarly, it also required huge execution risk, being able to build out a sequencing factory, do QC, be able to move samples through the system at scale. Next slide. And so when we think about what Broad is all about as an institute, it tries to bring together 3 capabilities. So first is the ability to identify large-scale, transformative, highly collaborative projects to take on. Then the second is creating new technologies in order to execute on those projects. And then finally, it's not enough to just create technologies, but it's also important to reduce them into practice and see them be able to operate at massive scale. And this is true for not only experimental technologies, so things like genome sequencing, but also for data technologies and data platforms, which is what I'm most excited about. Next slide. But what this means in practice is that having these 3 capabilities, we can then take on projects like the Human Genome Project and be able to make impact on society. But one of the things that I think is so interesting is that we're at this pivotal moment where the type of projects that we're actually taking on is starting to change. Next slide. Where the set of things that were once hard are actually becoming much easier, and the set of things that were once quite easy are actually now becoming much harder. Next slide. So when you think about the first decade of the Broad, a lot of what was hard was actually being able to generate data at scale. Over the last 10 years, we've generated something like 100 petabytes of genomic data. To put that in perspective, the entire movie corpus of Netflix is only around 2.5 petabytes. So this is big data by the scale of any organization. Similarly, it's not just about read-level next-generation sequencing, but we also have a slate of new data generation technologies. For example, if you think about something like single cells, we see a similar exponential growth in our ability to profile individual cells. And actually, in a widely cited paper from Nature a few years ago, written by Illumina, the main maker of sequencers, they pointed out that the rate at which data generation doubles in genomics doubles roughly every 8 months. So again, to put that in perspective, if you think about apps like WhatsApp or Facebook, they never doubled faster than every 6 months, even at their peak. So those of us who are working in genomics are seeing the limits of scalability as any industry has ever seen it. Next slide. But with this brings a new set of challenges. So genomics from its origins has always been quite good about data sharing. A lot of this goes back to the Human Genome Project, where the ethos was that the data should be shared by all. And so while we've been quite good about embracing the ethos of data sharing, our approach to operationalizing it has been a bit silly. In general, we put data on servers and we tell researchers to download it to their local environment. And this presents a slate of problems. The first is that in order to share data, you have to copy it, which means that you store a copy of every data set at every organization, which is huge cost to funding bodies like NIH or Wellcome Trust. As you start copying data and handing it around, it's very difficult to track and audit who's touched it and for what research purpose. Third, genomics is increasingly at risk of becoming a sport of kings, where to even begin to work with genomic and clinical data, you have to be at a well-resourced institute with a large amount of compute and storage and a security and compliance team. And then finally, we have a lot of need for elasticity. So what you're starting to see is a quite interesting moment in life sciences, when we're taking seriously the idea of inverting the model of data sharing. So that instead of bringing data to researchers, we instead bring researchers to the data by putting our data sets on public clouds where it's co-located with compute, and lots of people can work with the same copy of the data in a very secure and compliant environment. Next slide. So one of the flagship efforts at the Broad in this space is a data platform called Terra. This is developed jointly in equal partnership with both Microsoft and Alphabet. It's one of the first times that they've ever worked together on anything. And it already is home to data from more than 2 million participants in a number of flagship scientific initiatives. So today I want to talk about one use of Terra, which is very focused on our efforts during the pandemic and public health. Next slide. So since the beginnings of the pandemic, Broad has been very involved in a number of research initiatives to better understand the pathogenesis of this terrible infectious disease. And it's taken 4 basic phases. In the beginning of the pandemic, it was really focused on just getting the capability to sequence viral samples up and running at large scale, and as we sequence them, to be able to share them with the world through Terra. Then later, we started to use sequencing to be able to understand at a more detailed level the mechanisms by which the disease spreads through society. Then earlier in 2021, we started to see the rise of variants of concern, and the realization that just doing COVID testing with PCR was not enough, but actually doing much more widespread sequencing in order to be able to understand the spread of different strains of the virus. And then finally, really scaling up these efforts going forward to be able to try and build out a pathogen weather map of how the disease spreads through society. So let me just give a few highlights of what we've learned through this process. Next slide. Early on in the pandemic, right when in February of 2020, there was a now infamous biotechnology conference in Boston. This was right at a moment in time when many people wondered whether or not we should be pulling the plug on conferences like this, but they ended up proceeding. And what you see was from that one conference, a huge number of individuals ended up being infected, and because they were from around the world, they then carried it back to their homeland. During the early days of the pandemic in March, April, May, it was estimated that almost 2% of the cases of COVID in the world were actually due to this one event, and showed the impact of what a super spreader event could be. And of course, as we sequenced these samples, all the data and tools was made available to the researchers everywhere, nucleating collaboration across the scientific ecosystem. Next slide. Then more recently, we began to work very closely with the CDC, where suddenly there was the greater awareness of variants of concern and the need to be able to have public health labs start doing much more genome sequencing. For many of these public health labs, this was a new technology, and while they could buy sequencers, building out the informatics team was a much bigger challenge. So we partnered with them to use the pipelines that were already built in Terra and were easy to use by non-specialists. And again, for example, in the July 4th weekend, many of you were familiar with the Provincetown outbreak, which showed that there could be breakthrough infections in a way that we never expected. All of those samples were sequenced, and the data to build this evidence base was made available to the public. Next slide. And in fact, now we're working very closely with the CDC to start deploying genome sequencing of coronavirus across many different state public health labs. I'm showing a map of some of the early adopters right now. Next slide. So with that, I think this is a nice vignette on what collaboration looks like in a new world that's much more focused on remote data generation coupled with centralized analytics. And I just want to call out, during the past year, it's been a real pleasure to get to know the folks at BioCrossroads and Analytics Indiana. And we've been talking about a number of flagship initiatives focused on bringing clinical genomics into the care of patients across the state. And I just want to express how excited I am about these conversations and how much I hope that we'll have the opportunity to work together more deeply in the months and years to come. With that, I'll turn it over to the next speaker. Thank you so much.
Dr. Philippakis, thank you so much. Appreciate all of that and your presentation.